back with your next class. In today's class, we are going to discuss a very important part of this chapter, that is search for genetic material. So mainly in this section, we are going to discuss a few important experimental proofs. These experiments, they are going to prove that DNA is our genetic material. Alright. Now, even after DNA was known for a very long time, the DNA itself is our genetic material. That took a lot of time to get proven. Alright. So finally, these are some important experiments that were conducted by various scientists, after which later on it was finally confirmed that DNA itself is the genetic material. Always the debate was as per DNA versus protein, because there were a group of scientists who used to believe that protein, because it directly affects or directly rules our traits or characteristics, that's why maybe it is the protein which would be acting as the genetic material. It's not DNA, rather protein is our genetic material. But then later on, by various experiments, it was finally decided that no, genetic material or the storer of hereditary information is DNA and not protein. Okay, DNA controls our activity via proteins. Alright, but then the genetic material is DNA. Fine. So let's start with the first experiment, that is transforming principle. Now, transforming principle is first it was conducted by Frederick Griffith in 1928. Alright, in 1928, transforming principle was conducted by Frederick Griffith. Alright, do remember the name, it's very, very important. Now, experimental specimen that was used by Griffith for this experiment was Streptococcus pneumoniae, the bacteria that causes pneumonia. Alright, and for this bacteria there are two strains present. One is S3, another is R2. S here stands for smooth. So this is the strain of the bacteria that would produce smooth colonies in the culture media, and it is capsulated and hence virulent. That means it can cause disease. Whereas the other strain R2, here R stands for rough. Alright, so R2 strain of the bacteria, it is non-capsulated and hence non-virulent. So it does not cause any disease. Alright, now the experiment consists of 4 steps. It's a very simple experiment. The first step of the experiment is that live S3 bacteria, that is the virulent strain of the bacteria, is injected into a mice. Of course, because the bacteria used here, S3, is virulent, is disease causing, of course then the mice would die, isn't it? Next step is R2. Here live R2 bacteria, which is non-virulent, is injected into a mice. This time the mice would live because R2 cannot cause any disease and hence the mice won't die. Next, S3 bacteria, but this time it is hit-killed. Okay, hit-killed strains of S3 bacteria, it is injected into the mice. Now, if killed bacteria or dead bacteria would be injected into the mice, even if it is virulent, it would not be affecting the mice and the mice would live. Alright. But the last step is the most crucial one. In the last step, you can see a mixture of live R2 bacteria and hit-killed S3 bacteria together as a combination is injected into the mice. This time, see, R2 live, mice lives. S3 hit-killed also, mice lives. But when in a combination they are injected into the mice, this time the mice dies. And not only that, surprisingly, from this dead mice, Griffith could also extract a few live S3 bacteria. So when he observed live S3 bacteria in this dead mice, in the 4th step, he would conclude that something, okay, some substance, maybe we can refer it to be the transforming principle, it might have entered it might have entered from the hit-killed S3 bacteria inside the live R2 bacteria. And that's how that substance which entered, it transformed the R2 bacteria into S3. Understood? So that's how the R2 bacteria itself, it got converted or it got transformed into S3 bacteria. Alright, this is what was the conclusion drawn by Frederick Griffith after the experiment of transformation principle. But he did not further experiment, or he further did not try to investigate that what was the biochemical nature of this transformation principle. What was that thing that was causing this transformation? Alright, or rather what was the nature of genetic material? That was not experimented further by Frederick Griffith. Alright, he ended the experiment here itself. Much later, the biochemical characterization of transforming principle was conducted by three other scientists in 1944. See, it was in 1928 this experiment was conducted, and further extension of this experiment was done in 1944 by three scientists, Ostwald Avery, Colin MacLeod and Macklin McCarty. Alright. So these three scientists, they further tried to find out that what was the nature of the transforming principle. Alright. So for doing so, what they did is that they first extracted, okay, protein, RNA and DNA from the hit-killed S3 bacteria. Okay. They took a sample of hit-killed S3 bacteria, and from that hit-killed S3 bacteria of Streptococcus pneumoniae, they extracted three things: DNA, RNA and protein. Because any of this thing could have been the genetic material, which was not proven, which was not known. So all the three substances which could have been the genetic material were extracted. Alright. Now the experiment that they conducted was also very simple. What they did is that they took a few test tubes, and in those test tubes, say number 1, 2, 3 and 4. Okay. These test tubes were filled with liquid culture media, okay, or nutrient medium, where a bacteria could be grown or could be cultured. So in this liquid culture media, in all the 4 test tubes, they inoculated live R2 bacteria. Okay, live R2 bacteria is introduced or inoculated in all the 4 test tubes, and they were allowed to grow there, okay, for a while. After there was sufficient amount of R2 bacteria available in the culture media or available in the test tubes, now one after another, the DNA, RNA and protein of the hit-killed S3 bacteria were introduced in the test tubes, just to check which of the particle, whether it was DNA or RNA or protein, which might have brought that transformation, which could change the R2 bacteria which is already present in the test tube, which particular substance from the S3 bacteria could convert the R2 into S3. Okay, as simple as that. Fine. So in the first test tube, they introduced protein. Protein from the hit-killed S3 bacteria was introduced in test tube 1. And it was found that the protein could not transform R2 into S3. Okay. Next, they introduced RNA into another test tube, and further also they found that this also could not bring any transformation. Third was DNA. When DNA from hit-killed S3 bacteria was introduced in the test tube, this time it was seen that a few of the cells of R2 bacteria were successfully transforming into S3. So from this particular step, it was proven that DNA is the genetic material because it could transform the R2 into S3. Fine. But to further reconfirm that it was DNA that might have brought this transformation and nothing else but DNA, what they did is in the 4th test tube, they introduced DNA plus DNase. DNase is the enzyme that degrades the DNA. So when together, that means degraded DNA was of the hit-killed S3 bacteria was introduced into this test tube. Of course, degraded DNA would not be able to bring any transformation. So this further reconfirms that in the third test tube, whatever had brought the transformation was nothing but DNA. It was DNA which was responsible for the transformation to occur. Alright. So this was Avery, McLeod and McCarty's experiment, from where it was proven that for Griffith's transformation principle, it was DNA that would be bringing the transformation, and hence DNA is our genetic material. But then this experiment was not accepted well by most of the scientists, and that's why the search for genetic material would continue. Alright. Finally, in around 1952, it was two scientists, that is Hershey and Chase. They would conduct an experiment using bacteriophage, in which they would prove again that DNA is our genetic material. And from that experiment, which was well accepted, finally it was accepted well that yes, genetic material is DNA and not protein. Alright. So in the next class, we'll discuss regarding Hershey and Chase experiment. Thank you.